Thanks, Ashley, and thank you again for Bethany's wish for inviting me to talk to you about precision stratified medicine. So what's stratified medicine? Well, Kathy, I think, has already alluded to this. I believe that we've been practicing stratified medicine in paediatric oncology for years, and it's really where we apply a test, a biomarker, to select patients as to whether they should be higher risk, lower risk, and then to receive a treatment which hopefully means that they will have a very good chance of cure, but with the least burden, the least toxicities as possible. Whereas I think precision medicine is a different model, and of course there's been a lot of discussion about this, and there are some definitions. So here we see the US definition, which is discovering a specific unique therapy for an individual's cancer based on genetic analysis of their tumour. And this is the European definition, which is a model where you use molecular profiling to tailor the right treatment for the right person at the right time. And these are big things. So this is President Obama, who announced a precision medicine strategy in the US just 2 or 3 years ago. And even in the UK, our own Prime Minister was on the bandwagon in terms of the 100,000 Genome Project, which was about introducing whole-scale molecular genome testing in NHS patients. So where has this come from? Well, it started off, obviously, right at the beginning with the discovery of the structure of DNA in Cambridge, but really it started in the 90s with a project aimed to elucidate the human genome, so to actually have the sort of key to the code that actually codes the human body. And that went through, and due to international collaboration, was eventually actually drafted in 2003. The first genome was estimated to cost $3 billion. So obviously from that point of view, it was a major international effort, but you could see it was very difficult to understand how this could have practical significance initially. But something amazing has happened, which is technology in genome profiling has actually moved on so fast that from shortly after the Genome Project being able to sequence a genome for around about $100 million, we can now actually sequence it for $1,000, which is interesting because this Moore's law is actually computer technology. Now, certainly when I was a kid, my computer had a bat and a sort of square ball that you could play with, and now I've got my iPhone telling me things today. But actually, genomic sequencing has exceeded that technology advance. So you don't need to know why, but basically we used to sequence DNA molecules one at a time. Now it can be done of hundreds, thousands, millions at a time, which means that the whole process is, one, cheaper, but also is sped up, which is incredibly important. And there's a whole load of different ways of doing this, which are incredibly clever and complex, but actually are commercialized now. So this is actually something that's available in terms of researchers, but also diagnosticians. So this can actually be used in the clinic now. So there's been this sequencing explosion where as the cost went down, the availability to sequence genomes, and in our interest, cancer genomes, has gone up, and this has translated. And as Kathy alluded to, this has really changed the way that we think about childhood cancer. And in my own area of brain tumours, not only do we sort of have this molecular classification shown by Kathy earlier for glioblastoma, but we also have this in medulloblastoma, ependymoma, and also this rare children's, infant students, ATRT. And we now know that these are not all the same. You look under the microscope, you might think they're one disease, but they're actually subgroups. And within these subgroups, it allows us to look at the prognosis. So some children actually do better even with high-grade glioma. Some children actually will have a target that will be new. So it's allowing us to start to subdivide. Now that causes challenges because it means that rare tumours are becoming rarer. But of course, as Kathy's already told you, we're very good at collaborating in paediatric oncology across the world. So that's the way that we're solving this. So certainly, the American Society of Clinical Oncology believed this was the future of oncology, and they said that researchers and clinicians will have the tools to quickly conduct a panomic analysis of every patient with cancer. This was their bold statement. So is that actually truly going to be a reality, and what effect will it actually have for the patients and their families? Well, the idea is that the physician will just simply enroll a patient onto a study, or if this becomes routinely in the diagnostic lab, the patient will be genotyped, and that's in two ways: their tumour, but also, we'll talk about the issues of this, their germline. So what their genetic makeup, and be able to compare the two. This will actually be analysed, and this will come up with a list of targets and a list of drugs. So it all sounds very simple. And the ideal scenario is the patient will have some form of tissue. Now, that could be a tumour. The issue is that we know tumours evolve, and therefore this may mean that we have to start to consider difficult queries such as rebiopsying patients at relapse and continuously doing this to see how the tumour is changing and trying to evade therapy. Or it may be that we can start to rely on more surrogate markers. So we're investigating, looking at the blood, looking at the CSF in brain tumours to find tumour DNA that is sort of broken off from the main tumour and analyse that, which means it's much easier to do that, particularly for a child, to do a blood test or to do a lumbar puncture and get CSF. And then we analyse that, and then we get these readouts. So in the adult studies, as Kathy said, what you find is that a lot of tumours have a lot of mutations. And these are sort of already known, and therefore you probably don't need advanced precision profiling to find those, because we're already testing for those routinely. But what they found is that there is a tail in most adult cancers where there are rarer mutations, and some of those have matched drugs that you could try, but you might not think about actually trying those in most patients. So this idea of molecularly profiling and identifying these is very attractive. Now, in children's cancer, we have a lot less mutations. Now, that may be a problem, for instance, for immunotherapy, where actually some of those mutations develop antigens and we can attack them. But it may be in precision medicine for targeted therapies that that's helpful, because it might be that it's rather easiest to find the ones that are important driving the cancer. So I thought I'd give you some examples of how this has actually started to have an effect. And this is some first results in pediatrics coming from a couple of American institutions. So the first was from Boston and was using something called a targeted panel. And that's where you specifically look at genes that are already known to be involved in cancer. The advantage of this sort of system is that it's quite quick. You can use it on smaller amounts of tissue. And they performed their first study with 100 patients. It was both children and young adults. The average was 13 years of age. 89 of those 100 had sufficient tumour material to be able to sequence this, and 43% had a clinically meaningful result. But that does mean the majority didn't. And what that was in a third, there appeared to be a mutation that there was a FDA targeted drug or there was a clinical trial available. 6 of them had an actionable mutation, but unfortunately there was no matching drug to give a recommendation because it wasn't available. Now, very importantly, in 11%, although they didn't test the germline, the tumour looked like the child may have a cancer predisposition syndrome that could have been inherited. And that's also important because it might change your treatment. You may try and avoid certain things, but also the issue about genetic counselling, the impact for the family. But, and this is where I want to just temper everybody's enthusiasm for this, in reality only 3% actually received a matched therapy, and the reasons for it not being 30% was that the patient, by the time the results were back, their condition had deteriorated, or they'd moved on to a non-targeted therapy, or they was unable to get access to a drug. So at the moment, this is very interesting giving results, but actually really translating into the clinic, we're not quite there yet. Now, this is a different group. Now, what they did here was they actually did a much broader sequencing of the genome, and they also specifically sequenced the child's germline. And I think that this is interesting. So again, approximately 100 patients, very similar children and young adults. 89% there was adequate tissue. They focused a lot on what consent and being very careful about what people wanted to hear back with the results. And the majority gave consent to receive results. The turnaround time initially was 54 days, and that's too long to actually really influence it. They got it down to 48 days, but by getting this extra information and the computing power that you need to analyze it, there was an issue. It was also quite expensive. It was $6,000 per case because there was all these people that needed to employ. So although it might be $1,000 just to sequence, there was a lot of extra. But they did find 40% clinical meaning results. And in their case, because it was a much broader way of looking at the cancer genome, they estimated that in 15% it would change the treatment. It altered the diagnosis in some patients compared to what had been given previously. And it actually led to a change with 10% having a clinically meaningful benefit. So higher in this case because they actually sequenced more of the actual genome. Again, they found 10% of patients, this time directly, had a germline mutation, so there were implications in relation to that. And this has been followed up by the St. Jude's group, who specifically looked at germline mutations, and again, they found that children actually have 10% that there's likely to be a cancer predisposition syndrome, and in most cases that may be inherited. In some it will have started afresh. And in the majority of those cases, in 60%, there was no significant family history. So this was detected as a new finding, which can be both beneficial but have some challenges and some ethical dilemmas. So what are those? Well, at the moment, on our present data and knowledge from pediatric profiling, we know that 50% of cases will not have a clinical benefit. And I am slightly worried that everybody feels that when we do this testing, there will be an answer for their individual child, particularly at relapse. But we won't always find that. Now, if we do more in-depth profiling, hopefully we'll discover more. And looking at the epigenetics, we'll discover more, so it might be able to push that up. But at the moment, the reality is that 50-60% are not going to have a change. But there is this potential to change treatment, estimated at 15-30%, but we have to be aware there may be no mutation. The results need to improve, so the turnaround means that the child doesn't actually deteriorate while it's being processed. And of course, there could be a lack of availability of matched drug. But this big thing, that up to 10% may benefit. So the dilemmas are related to the feedback of results. Sometimes it may appear quite easy because the child will have a direct benefit. There'll be direct benefit for them, no. You could avoid damaging treatment, so perhaps there you would definitely want to know it. But in other cases, you need to think that testing that child's germline may have implications for other people, for the rest of the family, which means that they may be at risk of developing tumours or their children. And there can also be the issue that when you look at the whole germline, that you may find that that child has actually got a predisposition to something that may occur in adult life. And it could be something that you can't do something about at the moment. So it could be that they're programmed to get early Alzheimer's disease, a neurodegenerative or a cardiac disease. And that's got a big thing that if that child survives the cancer, that you've also got this information. So we need to think about the ethics and how we put that in place and include genetic counselling when we're giving these results. So does it work? I think there are very good evidence that this sort of practice is starting to work. This is targeting the BRAF gene. This is a trial that I've been involved with where we only give the drug to patients that have this particular mutation. And actually, rather than actually grouping it in terms of different tumours, we actually target it just on the mutation. And interestingly, that crosses across a big classification. So there's brain tumour patients, but there's also patients with a condition called histiocytosis. And what we're finding, and this has been expanded now, but this is holding out with much more patients, is that in relapse, we're seeing brain tumour patients having actual tumour shrinkage, significant tumour shrinkage, and durable, lasting over a year. Longest patients have been with relapsed high-grade gliomas have been on this for 3 years, but also in low-grade gliomas and histiocytosis, because it's the right drug with the right patient. And this has been shown by colleagues at Sick Kids as well, who've been looking at the response to standard chemotherapy versus targeted therapy. Down is good, up is bad, and unfortunately a lot of the patients with low-grade gliomas with this mutation don't respond well to chemo, but that with targeted therapy at the moment, all of their patients have. And this is one of my patients that I'm treating at the moment. So this patient's had 5 rounds of chemotherapy. They're very young, so we don't want to give radiotherapy. They've had previous surgery. They have a large brain tumor, so it's actually benign, but it's been aggressive. And this has been given dabrafenib, and this is just within 8 weeks. And the patient clinically improved, but there has been a massive reduction in size compared to the way chemotherapy would be. And this has been repeated on numerous occasions. So what's happening? I said President Obama has launched this. The adult study is open, and that's running. The pediatric study will be expected probably now, speaking with the guys there only last week, I think it will launch early next year. In France, they've been profiling, don't yet have a specific study. Lindley's going to tell you about the European initiatives for studies, but that's ongoing. They're moved from a targeted panel to this wider whole exome sequencing panel. In Germany, they've been doing the whole exome sequencing RNA-seq for some time, and it's been very impressive because they've been able to get their turnaround to 28 days, which is actually meaningful. In the UK, we have a targeted panel developed at the Marsden ICR, and this is being piloted as we speak at that centre, hoping to roll it out nationally very soon. And the 100,000 Genome Project is on board, and actually we do have a UK childhood cancer aspect to that, where we hope to be doing whole genome sequencing. I'm still slightly worried about the time it takes for this, but it should advance, and this is embedded within the NHS already. So in summary, I think that molecular profiling is a reality, it's here, and actually I do think it will become part of our standard practice. And I think that the precision medicine programs are being set up, and you'll hear that trials are being set up to actually benefit from them, as Lindley will talk to you about. But I think it's really important that we engage with patients and their families, and I'd quite like help in this, just to think about the expectations that are there, what are the ethics about rebiopsying, etc., but particularly how we feed back the results, because this is a bit of a can of worms. It opens a lot of issues, not just for the patient, but for their immediate family. And we need to think about how we deal with that. So I think there is massive potential, but we mustn't overestimate in terms of it giving an actual direct therapy. It's going to improve diagnosis, and I think it's going to learn a lot from the science. But at the moment, there will be a select group of patients who will really benefit from it, but there may be many at the moment that wouldn't be the answer, but it may be in the future. So unfortunately, I can't stay for the workshop later on, but I had a few questions of my own, which actually I'd quite like to get help from the community. And I think this could be a project, probably a simple one, might not even need funding, where we actually get a group of parents and a wider group to help with some of these questions. You don't have to go through them, to feedback. And I'd be really keen to work with people in a group on looking at that. And I think we're going to take questions after Lindley's talk. Thank you.